we started to look at the structure of nucleic acids, and particularly we were looking at the structure of the monomers of nucleic acids, which is the nucleotides. So we saw the components of the nucleotides being the pentose sugar on which we have the nitrogenous base and the phosphate group. Then we even looked at some nucleotides that are not present in nucleic acids in addition to the ones that we find in nucleic acids. We'll continue today on that, looking at now bringing the monomers, which are the nucleotides, now bringing them together to form a nucleic acid chain. So we are continuing with more complexity. And then we'll end it on structure today. The next time when we meet, then we continue to look at function of nucleic acids. We can have a brief overview of the function of nucleic acids, but then next week when we meet, then we continue to go into the details of that. So let's continue from where we left off last week. And today we're starting with what we refer to as the primary structure of nucleic acids. Now you remember you refer to proteins made up of amino acids. And you also remember that these amino acids are bonded together. So if you already have knowledge about these proteins, the amino acids, the different amino acids that you have, glycine, leucine, isoleucine, phenylalanine, and all of that. And you take a peptide chain, or else you take a protein, and you are referring to the structure of the protein by referring to the sequence of the amino acids that follow. So for instance, if you say that glycine is bonded to isoleucine, which is bonded to phenylalanine, which is bonded to tyrosine, and you continue to say that, you are talking about the sequence of the amino acids that bind together. When you are doing that, what you are doing is you are describing the primary structure of the protein or the peptide. The same way, acid chain or in a nucleic acid strand, you are describing the primary structure of the nucleic acid. So the primary structure of nucleic acid is just about the sequence of the nucleotides. Now, when you look on your screen on the left, you have a fragment of a nucleic acid. Now, we already know the structure of the nucleotide to be composed of a sugar, which is represented here by S, the letter S. And on that sugar, you have a base. And on that sugar too, you have a phosphate group, which is represented by the letter P. Now, you see that the bases are represented by different colors, and then there are some letters that are written in them. Those letters are the first letters of the names of the bases. that are on the sugar. So we learned these nitrogenous bases, purines and pyrimidines. And the purines we saw were adenine and guanine. So when you see gene refers to guanine. When you see A, it refers to adenine. Then the pyrimidines, cytosine, thymine, and uracil. So when you see T, it refers to thymine. When you see C, it refers to cytosine. Now here, you don't see uracil. That is because this is a fragment of DNA. And it's a DNA strand. And in DNA strands, you don't find uracil. In their place, you have thymine there. So that's why you don't see uracil here. Now, what you notice is that you have some repeated sugar with phosphate group on a sugar phosphate. That is forming what we refer to as the backbone of the nucleic acid strand or the nucleic acid chain. You see, it's the same. It's just sugar, sugar phosphate, all of this. So you see it even being depicted here. P is the phosphate group, S is the sugar, and it's repeated there. So that's a backbone of the nucleic acid. And it is only the bases that are different. So the bases, we show them with different colors and even the letters of the bases, of the names of the bases in them to distinguish these bases. As a matter of fact, it is the bases that you use to distinguish between nucleotides. Because as you can see clearly, the sugar and the phosphate group is constant in all the nucleotides. So it is the basis that you use as a distinction between the various nucleotides. So this nucleic acid strand, which you are seeing, if we are writing it or if we are describing it, that this strand is composed of guanine bonded to guanine, bonded to another guanine. So gene. So guanine, thymine, guanine, adenine, cytosine, thymine, cytosine. Now we are referring to the nucleotides just by the basis because it is the basis that give the distinction. So when we say guanine, what we actually mean is guanosine monophosphate. And when we say thymine, what we mean actually is thymidine monophosphate. When we say adenine, what we mean is adenosine monophosphate, because you know in the nucleotide, what you have there is a sugar on which you have the base and the sugar, the base and the phosphate group bonded on the sugar. So really, when we are saying guanine, we don't just mean the base, we mean the nucleotide, because it is the nucleotide that is binding to form the nucleic acid chain. So as we describe that, guanine, thymidine, guanine, adenine, cytosine, thymine, cytosine, we are referring to the primary structure of the nucleic acid chain or the nucleic acid strand. So the primary structure of nucleic acids is just the sequence of nucleotides that are in a nucleic acid strand. Now we are talking about nucleic acids composed of nucleotides that are bonded together. So then we start to talk about, so what is the bond that binds the nucleic acids together? You see, the same way we say that, say, carbohydrates are made up of monosaccharides. And the bond that exists between the monosaccharides in a carbohydrate is a glycosidic bond. And when we take a protein too, we say that the protein is made up of amino acids. And the bond that exists between the amino acids is a peptide bond. Now we take a nucleic acid. And a nucleic acid is made up of nucleotides. So these nucleotides bond together to give you a chain, a strand. What is the bond that exists between the nucleotides? That bond is what we are referring to as a phosphodiester bond. Now, you remember that when we were looking at the structure of the nucleotide, I mentioned that the bond that exists between the phosphate group and the sugar is a phosphoester bond. And I told you to keep that in mind because we'll come back to it. So this is where we are coming back to it. Now, on the right-hand side, you see a more detailed fragment of DNA. This time around, it's not like what we saw previously, that we just show SS like for sugar, PP for phosphate, and then we drew some shapes and wrote the letters in it to represent the nitrogenous bases. But we are actually seeing the full structure of the nucleotides here. So we see, starting from the top, we see a full nucleotide with a base adenine on the ribose, the deoxyribose sugar. And that sugar binds to the phosphate group of the next nucleotide, on which you have thymine. And you see that nucleotide also binds to the next nucleotide that's having the base guanine. And then it binds to the last, which is at the bottom, which is with the base cytosine. And you see between the nucleotides, all the bonds are shaded in orange. You see all the bonds are shaded in orange. Now, those bonds are what we are referring to as the phosphodiester bond. Why is it a phosphodiester bond? Why is it called so? I just said the bond that exists between the phosphate group and the sugar in a nucleotide is a phosphoester bond. A phosphoester bond because an ester will have a carbon. So in place of that phosphorus atom, you have a carbon on which you have a double bond, O, oxygen, and that carbon will bind to an oxygen, and the oxygen will bind to another carbon. That group will be called an ester. But when you replace that carbon with phosphorus, then you now have a phosphoester. So if the bond that exists between the phosphate group and the sugar is a phosphoester bond, and you pay attention to the bond that is existing between that phosphate group and the nucleotide above it that it is binding to, you will see that it is the same type of bond that you are having between the two nucleotides, the phosphate group and the nucleotide that is above it. So you see that the nucleotide is binding to carbon number 3 on the sugar of the nucleotide above it. So that is carbon that is binding onto oxygen. And that oxygen binds to that phosphorus, which is having a double bond oxygen on it. So that is another phosphodiester bond. So now you have two phosphodiester bonds between the nucleotides. That's why we are calling it phosphodiester bond. Phosphodiester bond. Now, another thing that I want to draw your attention to is the fact that you see when you write the fragment of a nucleic acid with those letters of the bases in the nucleotides. So for instance, if you take this fragment, the fragment which is on your screen now, and we are writing the fragment just using the first letter of the name of the bases that are in the nucleotide from the top coming down. It means we will say A for adenine, T for thymine, G for guanine, and C for cytosine. So we'll write ATGC. If you are just writing that primary structure with the letters. But you also see that when we write these fragments, we usually write, maybe at the beginning you write 5 prime. And then at the end you write 3 prime. What do we mean by that? Now, look at the nucleotide above, the first nucleotide with adenine on it at the top. You see that the carbon of the sugar at the end there is 5 prime. There's a 5 prime where if we should be binding another nucleotide to this last nucleotide at the top, if we should be binding another nucleotide to it above it, it will be binding on this carbon number 5. Because you take note that the phosphodiester bond is between carbon number 3 of the nucleotide that is the first nucleotide with adenine and carbon number 5 of the nucleotide that is below it, thymine. And you see all the bonds are between carbon number 3 and carbon number 5. Carbon number 3, carbon number 5, carbon number 3, carbon number 5. So you see that the last nucleotide at the top is ending with the carbon number 5. And the last nucleotide, which is below, the last one down, you see the sugar there. The open carbon atom onto which the next nucleotide can bind to is carbon number 3, which is also numbered there as 3 prime. So if we want to write this, fragment with those letters and show the ends for this fragment, which is here, I'll write 5 prime, A, T, G, C, 3 prime, to show the ends of the fragment that we are talking about. You can write it the other way too, like from down to the top, where you say 3 prime, meaning this is the end, 3 prime, C, G, T, A 5 prime. It's the same thing. So now when you see those numbers 5 prime, 3 prime, you should know where it's coming from. You should understand why it's written that way. Then you also will understand that the bond which is existing between these nucleotides is the phosphodiester bond. And the phosphodiester bond exists between which carbons? Carbon number 3 and carbon number 5. All right. Now, we move on to what we refer to as base pairing. You see, so far we were talking about nucleotides that are binding to form a chain. And we say the bond which is existing between these nucleotides is a phosphodiester bond. But when you take DNA is double-stranded. It's having 2 strands of nucleic acids in them. Now, these 2 strands also have bonds between them. And this bond exists between the bases of the nucleotides. Now we have what we refer to as the Chargas rule. And this is coming from this American scientist, Edwin Chargaff, but actually he's having an Austrian Hungarian. I think his roots are from Austria and Hungary, but he was in the United States working as a biochemist. And one of his works demonstrated that when you take a DNA fragment, the amount of adenine which is present in that fragment is equal to the amount of thymine which is also present in that DNA strand. And similarly, the amount of guanine is equal to the amount of cytosine which is present in that fragment. So then, the conclusion was made that since you have DNA which is double-stranded, it meant that the bases which were having bonding between them were in pairs, such that you have adenine binding to thymine, and you have guanine that is binding to cytosine, which is why you have their amounts being equal. Now, that also means that in base pairing, a purine should be binding to a pyrimidine. So you don't have a purine binding to another purine. Like we studied the nitrogenous bases, the purines were adenine and guanine. You don't have adenine pairing with guanine, which is another purine. And the same way, you don't have cytosine to be pairing with thymine. Both of them are pyrimidines. Of course, uracil, when we move on to look at, say, maybe secondary structure and above of RNA. Yes, there can be some pairing in RNA as well, but that will not be in a double strand. So the Chagas rule simply is stating that in a nucleic acid strand, that is specifically DNA, you have a purine pairing with a pyrimidine. But then you also keep in mind that that rule is also telling you that adenine pairs with thymine and guanine pairs with cytosine. So it's not like any purine at all is binding to any pyrimidine at all. So for instance, it's not like if you take adenine as a purine, it doesn't mean that because cytosine is a pyrimidine, adenine is going to bind to it. No. Because we have said from the beginning that the amount of adenine was found to be equal to the amount of thymine. And the amount of guanine was found to be equal to the amount of cytosine. And so the conclusion clearly was that adenine will always pair with thymine and guanine will always pair with cytosine. All right, so that understood, we move on to look at how these bases pair. We said the bond that existed between the nucleic acids was a phosphodiester bond. That's in a strand. Now, when you have a double strand and you are having base pairing between these strands, what is the bond that will exist between the bases, between the strands? That bond is a hydrogen bond. So hydrogen bonds will hold the two strands together. So if you take a DNA molecule, you have phosphodiester bond, which is holding the nucleotides in one strand, in a single strand. And you also have the hydrogen bond, which is holding the strands together. So these are two different bonds. Now, when you take adenine and thymine, we see here being shown that adenine and thymine have two hydrogen bonds between them, two. And these bonds are shown in the dotted red lines. So the number of hydrogen bonds that exist between adenine and thymine is 2. Now, how many hydrogen bonds exist between guanine and cytosine? Also being shown here in dotted red lines, they are 3. 3 hydrogen bonds existing between cytosine and guanine. Now, this brings to what we refer to the term complementary base pairing. So, what does it mean when you hear complementary base pairing or complementary base pairs? It only refers to the fact that adenine pairs with thymine and guanine pairs with cytosine. So what it means is that adenine is complementary to thymine, or you can say adenine and thymine are complementary base pairs. The same way, guanine is complementary to cytosine, and vice versa. Cytosine is complementary to guanine. So these two are also complementary base pairs. So when you hear the term complementary base pairing or complementary base pairs, this is what it refers to, that you have adenine that pairs with thymine, and you have guanine that pairs with cytosine. Right. Now, we can continue to look at more complexity of the structure. We've talked about the primary structure of nucleic acids being the sequence of nucleotides in a nucleic acid strand. And that is so for both DNA and RNA. Now we take the secondary structure of DNA, which we refer to as a double helix structure. Now, in first year, when you did biological chemistry, I believe you did structure of proteins, and you saw secondary structure of proteins, which was an alpha helix. That was a single peptide chain, which was winding like a ribbon, having that right-handed wind, twining up. Now you are having a DNA which is double-stranded. And these double strands, so we see here again, we see one strand is being shown in blue. And the second strand is being shown in orange or red. And these two strands are winding around each other, up, winding, so forming a double helix structure. And you see between these two strands, we see being depicted the base pairs, like you see thymine pairing with adenine, cytosine pairing with guanine. We see that being shown throughout. That structure is what is being referred to as the double helix structure. Now, another thing is that when you have such double helix structure, that twining of the DNA, you do have some spaces that are small, some are larger, like there are some gaps that are formed between these winds, which are referred to as the major groove and the minor groove. Usually, these spaces, when we come to look at it, I think in this course, we will come to some what we refer to as protein motifs. So yeah, this is a nucleic acid, but this nucleic acid forms complexes with proteins. So you have some regulatory proteins that can bind onto this nucleic acid, controlling its function. And this will usually be binding in these spaces, the major groove and the minor groove. So the major groove will be for like some larger protein molecules. Those that will bind to minor grooves will be, smaller protein groups that will be binding in those minor grooves. I mean, when you read generally, you will say that you have the minor groove that is consisting of about maybe 5 to 6 base pairs, and the major groove is like double of that, like you have about 10 to 12 base pairs of that space that you find over there. Another thing that you can also keep in mind is the fact that you see we were referring to the 5' and the 3' ends. So we're looking at just one strand. Now, when you take two strands, what happens is that you have one strand. If you are looking at the strand from top down, one strand will be running like from the 5' end to the 3' end, whilst the other one will be running in the opposite direction. So from 3' end to the 5' end, in order to bring the bases in close proximity to each other to have hydrogen bonding between them. And because of that, you also see the term anti-parallel directions, or just say opposite directions. These two nucleotides, the two nucleic acid strands, are running anti-parallel to each other. So that's what that also means. Means you can even just draw, two straight lines, parallel lines, and then you have 5' written at the top, 3' down for one, and the other one, the opposites, 3', 5'. So just to show that these strands are running anti-parallel to each other. Right, so that's about the secondary structure of DNA. Now let's take a look at more complex structures of the RNA molecule. We have known the RNA to be single-stranded. So I think all along, even in secondary school, you've known that the DNA was double-stranded, but RNA is single-stranded. So when we talk about a secondary structure of RNA, what really do we mean? Now, you have RNA, which is a single strand, but that single strand can fold. And when it folds, it is possible that at some positions, you have complementary base pairing. The only thing is this time around, this is RNA, and so in place of thymine, you are going to have uracil. You keep that in mind. There are various secondary structures of RNA that can form. The first we are seeing here is what is being referred to as hairpin, the hairpin structure. And you see the hairpin structure, it's called like that because, usually the ladies have this hairpin, the pin they used to hold the hair. It looks like it. So they call it a hairpin structure. Now, what do you see? You see this is an RNA fragment that, folds. And when it folds, we see there's a portion which is violet being shown, like there's some hydrogen bonding between some bases there. We see it being shown more in the next, where we are seeing some stem loop. So we see that an RNA folds at a portion at the top. There is no base pairing; it's just open up there. But then you have a place where what we are referring to as a double helical stem region is having some base pairing between the strands that are close, with the bases there having the hydrogen bonding between them. Now, you can have the forms of RNA secondary structures being referred to different names, like a bulge, for instance. And when you look at it, what it means is that you are having a stem, stem region where you have some complementary base pairs, and then along the line, you are having some uncomplementary, base pairs that bulge out of that stem, and then right after that, you have complementary base pairs, and so the stem continues. That we refer to as a bulge. So it's bulging out. And next to it, we also have what we are referring to as a single base bulge. So when you look at what we refer to as a bulge before that, so it means you can call this a multi-base bulge, because you have more than one bases that are bulging out. But here you have only one base that is not binding to any complementary base opposite it, and it's called a single base bulge. You have another one that we are referring to as a mismatch. Now, a mismatch, because you see here too, you are having a stem. You have these complementary base pairs with hydrogen bonding between them. Then along the line, you have bases. You have two bases that are opposite each other, but they are not complementary, and because of that, there is no hydrogen bonding between them. But right after that, you have the stem continuing. That is referred to as a mismatch. Sort of also looks like a bulge, but this time around this is due to the fact that you're actually having bases there opposite each other, but they are uncomplementary. So they are mismatching. That's why you don't have the hydrogen bonding between them. But in the case of bulging, you see you don't have any base at all opposite the base that has bulged out. In both cases, the multi-base bulge, you don't have any base on the opposite side. Single base bulge, the same. You don't have any base opposite it. That should be binding. It's just that single base that bulges out. You can also have what is being referred to as a symmetric internal loop. I mean, when you look at it, it means you can even say this mismatch is almost like you can also say it's a single base symmetric internal loop, because it's the same kind of thing, that you have a stem, that you have a number of bases along the line that are uncomplementary. And so you have, here it's being shown that there's 3 on the left-hand side, 3 on the right-hand side. It can be any number, but they should be equal on both sides to make that loop symmetric. And asymmetric, the last one you are seeing is asymmetric internal loop. That asymmetric internal loop, you see that you have a number of bases on the left side, and then you have just one base on the right-hand side. So on both sides, you are not, the number of bases that are bulging out are not equal. So it makes it asymmetric. So that's an asymmetric internal loop. That can be any number. It can be 3 against 2, 4 against 1, 4 against 3. Once they are not equal, it's going to be an asymmetric internal loop. So you can have these different forms of secondary structure of RNA. Now, when you have an RNA molecule that is having more than one secondary structures in it, what you have is a tertiary structure of RNA. So the tertiary structure of RNA is an RNA with more than one secondary structures in it. So for instance, what we see here, pseudoknot. See a pseudoknot, what is being referred to as a pseudoknot. And here we see an RNA strand, 5' end, and it goes up, loops, comes down, and then it loops up again to the 3' end. And what we see is that at certain positions, you have what is being referred to as stem 1, which is depicted in green, showing that there is bonding between some complementary base pairs that are close to each other. But you see, this is just one strand. It's not like DNA that you are having two single strands that are opposite each other, that are having that hydrogen bonding between them. It's only a single strand, but that single strand is folding. It has folded. And because it has folded, you find some bases that are complementary being close together and having that hydrogen bonding between them. And you see there are two stems. The first stem is shown having that green color. And the second stem, when you have the strand looping up, you see a second stem that is labeled stem 2, which is having the violet color, also having hydrogen bonding between complementary base pairs. So what it means is that in this structure, this pseudoknot, you are having two secondary structures. And because you are having more than one secondary structure, this pseudoknot will be said to have a tertiary structure. It's the same thing which is being shown next to it, this time around giving more detail. So the green portion where you have stem 1 is shown now with the nucleotides that are complementary and having the hydrogen bonding between them. So you see guanine, cytosine, all the way up. You have, okay, here you see some uracil, adenine. So remember, it is an RNA molecule. So in place of thymine, you are seeing uracil. Then you see the loop 1 portion where you don't have a complementary base pairing is shown in red with some bases there that are not complementary to the bases opposite it. So there's no bonding between them. So no stem is being formed. Then you see the violet region, which is depicted as stem 2, is also being shown with some complementary base pairing over there. And then you have loop 2 here, which is also red, showing some uncomplementary bases here that are not having any hydrogen bond with the bases opposite them. So this second one actually gives more detail than the first one. The first one is showing, stem region with like a ladder, showing those hydrogen bonding that can occur between the loops. But the second one here is giving more detail, showing the bases that are paired and having the hydrogen bonding between them. The third one really is, it's just some kind of a drawing, also showing you can have this type of folding in RNA molecules, showing the loops and the stems, sort of giving some cyclic structures at the center there to show that those are bases that can interact between them. But these will not actually give you much information as far as chemistry I mean, the biochemistry is concerned, but it gives some pictorial representation of what we are referring to. You find a common tertiary structure, secondary tertiary structure you find in RNA will be in transfer RNAs. So if you go looking at the structure of transfer RNAs, so RNAs that are responsible for transferring amino acids one after the other onto the messenger RNA to bind them to form a protein in a protein synthesis process. The transfer RNA is having such tertiary structure. You also find tertiary structure of RNAs in, we have some catalytic RNAs, like, usually we have proteins that are enzymes. Like enzymes are mostly proteins, but you do have some RNAs that also act as enzymes, and mostly they will be acting in the metabolism of nucleic acids themselves. So they act in the breakdown of nucleic acids, because nucleic acids undergo some recycling process where you can have the nucleotides. First, the nucleic acid is broken down into the nucleotides, and even the nucleotides broken down into the various components, and it goes back into synthesis of new nucleic acids and all of that. And you find some RNAs that act as biocatalysts, like enzymes, in these biochemical processes. Some of them are what we are showing over here as leadzyme. So you see it's having some secondary structures. So like different subunits of secondary structure RNAs that come together forming a complex. You see one, the hammerhead ribozyme and the twister ribozyme. When you look at them, they look much like a DNA molecule. You might think that it is a double helix structure. But no, it's not a double helix structure. It's an RNA that is, winding, looping, and forming that helical stem loops. But it's not double helix. It's not coming from two strands. So it's not a double helix. Even it's a helical structure, but that helical structure is coming from only one strand that is folding and giving you, such structures. So you know that there can be secondary structures in RNAs as well, and there can be tertiary structures. Then there can also be quaternary structure of RNAs. So it's just like when you were studying proteins, you refer to the quaternary structure of proteins, where you have proteins with subunits that come together to form a molecule and be acting as one. A common example you refer to is hemoglobin, which is made up of 4 subunits, 2 alpha subunits, 2 beta subunits, and these come together to give you the whole molecule of the hemoglobin. You can have a different number of subunits of proteins that come together in different types of proteins. So usually you have things like dimers, which will be made up of 2 subunits. You have some trimers, tetramers. So hemoglobin will be a tetramer, a pentamer, all the way hexamer. You can have them. Complexes of proteins, different subunits, they come together and they are acting as one molecule, carrying out a single function or maybe multiple functions. And that will be referred to as a quaternary structure. The same way in RNA, you can have RNAs with tertiary structures, which are like monomers, and they are coming together to form a complex to carry out a specific function. That's a quaternary structure of RNA. Even though you see it to be complex, it's easy to distinguish the monomers because you can see like some repeated units. So you can quickly identify that this portion is a monomer of its own. This is also another monomer of its own, and they are coming together to act as one complex to carry out a specific function. So you can have these complex structures of RNAs as well. I hope the information is not getting too much. Now, you see at the beginning, I said that for biochemistry at all times, you realize that when a component is taken, the structure of that component is studied. What is the component made up of? Like, what is this structure made up of? If you take protein, you say, what is it made up of? You go and look at the amino acids, the various amino acids, and then what type of bond that is between them. So you look at all of that structure, and then later you start to talk about the function of the protein. So it's the same thing which is happening here, that we take a nucleic acid and we look at the structure. What is the nucleic acid made up of? So first we say that nucleic acids are divided into RNAs and DNAs. Then we say each of these nucleic acids are made up of nucleotides. And then we start to look at the nucleotide itself as a monomer, a single unit. What is it made up of? We look at the bases that make up the nucleotides, the sugars. The phosphate group is the same for all of them. We studied all of that, and even now went on to look at the nucleotides binding together to give you a nucleic acid chain, and even further look at more complex structures, secondary structures for DNA. Then for RNA, we even went further, look at secondary, tertiary, and quaternary. Now we are moving to function of DNA and RNA. So function of nucleic acids, that's what it means. And I mean, what's the major function of nucleic acids? We said it right from the beginning that nucleic acids are storage forms of genetic information. So they store genetic information in a cell, and they transfer that genetic information to daughter cells. So the nucleic acids are responsible for the transfer of genetic information from parents to offsprings. Generally, that's what we said. This is just an overview of what really the process of transfer of information, that genetic information is. Next week, when we come, we'll look much more into detail to each of these processes, but we can take an overview of this thing today. And then when we come next week, we can go ahead and look at the specific processes. We take one after the other. And what are these processes? Now, there are three major processes. The first is replication. Replication. So this is where a DNA molecule, out of one DNA molecule, you have a new molecule being produced. So a one DNA molecule yields a new DNA molecule. So it's more like a DNA molecule giving birth to another DNA molecule. So we'll look at that process of replication, how that occurs. Now, where does the DNA exist in the cell? Let's take eukaryotic cells, because eukaryotic cells have a distinct nucleus. And we know that the DNA is in the nucleus. The DNA is in the nucleus. So it means that the genetic information is stored in the nucleus. Now, that genetic information, which is stored in the nucleus, has to be copied first onto a messenger to send to the cytoplasm for that genetic information to be translated into something. Now, that process of copying the information from the DNA is what is being referred to as transcription. And what is it copied onto? It is copied onto what we refer to as the messenger RNA. So the messenger RNA will carry the information from the DNA in the nucleus and bring it to the cytoplasm. It's a messenger. So messenger RNA is bringing a message. It's bringing a code from the DNA in the nucleus into the cytoplasm of the cell. Now you see that process of copying the information from DNA onto a messenger RNA is what is being called transcription. You are transcripting the code, you are copying it. Then the messenger RNA, when it comes into the cytoplasm, the code has to be translated. That process is what is being referred to as translation. And how does that information get translated? You see the messenger RNA, when it comes into the cytoplasm, you have a ribosome complex. So here again, ribosome is one of those organelles also you studied. I think you saw ribosome even in the secondary school, and I'm sure you saw ribosome in first year biochemistry. So the ribosome comes to bind to the messenger RNA. It's a complex. And then you see also transfer RNAs are binding to amino acids. So you see amino acids that are shown in some violet spheres. So violet spheres that are bonded on transfer RNAs. All of these is inside the cytoplasm. And these amino acids are then coming into the complex that the ribosome forms with the messenger RNA. And what do you see? You see that the amino acids, which are the violet spheres, are joining one after the other. They are joining. And in the end, what you are having is what is being shown here as a growing peptide chain. So a protein is being produced by the joining of the amino acids that the transfer RNAs are bringing onto the complex. And in the end, you have a protein that is synthesized. So that protein is actually the message. Like that is the translated message. The code, when it is translated, becomes a protein. And what's a protein? A protein can be an enzyme, it can be a hormone, it can be a neurotransmitter. So for instance, when you have a particular reaction which is supposed to occur, there's a requirement for it, which is that there must be an enzyme. Maybe there should be a synthesis of hemoglobin, or let's say the heme, only the heme, for instance. Or there should be synthesis of a certain hormone to modulate a particular reaction. And an enzyme is needed. And when an enzyme is needed, then you see there is a trigger of that DNA, a particular genetic code, a gene, which is supposed to be expressed. Actually, we say the process of synthesis of protein from the DNA. So from all this process where you have transcription and then you have translation, where you are having the protein out of it, you refer to all of these as gene expression, protein synthesis. The gene is being expressed and you have a protein being produced. So that protein then now becomes the hormone that will go and regulate the particular reaction. Step by step, these things occur. So what we'll do is, in order not to mix all of these, the function, if you want to add it to the structure, it will be too much to take at a time. I give you this overview of what happens with the function of nucleic acids, where you have these three major processes: replication, transcription, and translation. Replication, if you think of it, is not only about the formation of a daughter RNA from a parent RNA, but it can also mean opening up of the DNA strand in order for it to be copied transcription. So apart from the fact that we are seeing the replication being at one side, which is particularly for the production of a new strand, or having a new DNA molecule from the old one. You can also have just the opening up alone. You are, to have the process of transcription sometimes can also be referred to as replication. But mostly, when you say replication, what it means is that you are replicating, you are reproducing one RNA, one DNA molecule, you are reproducing it so that you can have a new DNA molecule out of the old one. We'll see that process as we continue looking at the function of DNA and RNA. Then I think, okay, let's take a look at the genetic code. Then we can end it here for the genetic code. And then when we meet next time, we will then start looking at the processes one after the other. Now, what we refer to as a genetic code is, you see, remember, the messenger RNA is bringing an information from the nucleus to the cytoplasm. That's a code. And what is that code? Those are nucleotides. They are nucleotides. And we have seen the nucleotides, we represent them by the first letter of the basis of those nucleotides. Now, the genetic code is made up of triplets, like you have 3 nucleotides that code for a particular amino acid. And we see this is being shown such that you have on the left-hand side, vertically, you have the bases being shown: U, uracil; C, cytosine; A, adenine; guanine, G. that will represent the first nucleotide in the triplet of that code. Then a second nucleotide in the triplet is being represented by these letters horizontally at the top. Also the same thing, uracil, cytosine, adenine, and guanine. And the third nucleotide in the triplet is what you see vertically represented on the right-hand side. So we see first letter, second letter, third letter. Now what you see is that each of these codes, like if you say uracil, 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 and even uracil, uracil, cytosine, each of them is coding for some specific amino acid. Actually, all of these codes, I think should be giving you like 64 of these codes, but only 61 of them are active because, of course, you are seeing that you are having some of them which are stop codons. We'll come to that. So you have 61 of them that will actively code for an amino acid. So we see, if you see PHE, that is phenylalanine, LEU, leucine, ILE, isoleucine, VAL, valine, SER, serine, PRO is proline, THR is threonine, ALA is alanine, TYR is tyrosine, HIS, histidine, GLN, glutamine, ASN, asparagine, LYS, lysine, ASP, aspartic acid or aspartate, GLU, glutamate or glutamic acid, CYS, cysteine, TRP, tryptophan, ARG, arginine. And I think we saw this SCR already, serine, ARG, also arginine. And then you have GLY, glycine. So these are, the different amino acids which is being shown. But this is what I want to say. This is not meant for you to cramp in your head, please. There's no point cramping these, codes like you say. I mean, nobody will be asking you that, like, I just come and I give you CCU, which amino acid will it code for? No, you don't do that. So please don't cramp this. Even if I give you any question, concerning this, it will be in a form of, say, I give you maybe a messenger RNA strand, like I give you the sequence of the nucleotides in the messenger RNA strand. And I am asking you to give me the amino acid sequence of a peptide that can be produced from that messenger RNA. But even that, I'll provide you with a genetic code. This table should be there so that because this table is a reference that you should just refer to. Like you are having a sequence of nucleotides and you want to determine the amino acid sequence that can, be synthesized from that RNA molecule, that's messenger RNA. So you first of all know that the genetic code is in triplets. So you first, mark the triplets then you begin to look at each of those triplets and the corresponding amino acids that you find in the genetic code table. And that is how you can determine such amino acid sequence. But you don't go cramping this into your head. No, I mean, please, nobody should waste his or her time doing such a thing. Another thing you can pay attention to, maybe these ones, yes, you can remember them. Like you have one of the codes is being shown in red. AUG. AUG is coding for methionine. You remember methionine was one of the amino acids with sulfur in it. And the second was cysteine. AUG, you see, is red because that we refer to as a start codon in the process of protein synthesis. AUG is the code that starts the protein synthesis process. And what it also means is that the beginning of the peptide chain, which is synthesized from the messenger RNA, is always methionine, because that is the amino acid that code AUG, codes for. I mean, it should be easy to remember. Just AUG looks like a short form of August. So when you remember August, then you know that is the code for methionine, the start codon. Then we have three stop codons. These don't code for any amino acid. That's why they are referred to as the stop codon, which means for any gene that is coding for a particular protein, it will end with these codes, so that when the process gets to these codes, no amino acid joins, and so the protein chain dissociates off from that ribosome messenger RNA complex. And these ones should be easy to sort of remember. They are all starting with uracil. So uracil, adenine, UAA is a stop codon. And these two, UAG, UGA, they are also stop codons. The same bases in there, adenine and guanine, but if you change their positions, so you either have UAG or UGA, they are also stop codons. So you have three stop codons and you have one start codon. So these ones, yes, you can pay attention to them. You can easily remember them, but you don't go cramping the whole of this into your head. Your brain is an important organ that you should keep to do more important things than just cramping things in it unnecessarily. All right, so I will stop here and I will open the floor now. If you have any questions, any contributions, any concerns, I'm monitoring the chat. No one has written there yet. You can write there, or you can raise your hand and I will enable your microphone. I'll call your name. You unmute yourself and then you speak. So the next time we'll continue with protein synthesis. All right, I see two hands up. Ebenezer, let me call Ebenezer first. Is the one at the top. Ebenezer, your mic is allowed. You can unmute yourself and speak.